The slide deck that I'm using today is titled Week 9, Day 1. It's also known as Sugars Part 2. Um, so I'm going to share my screen so that you guys can see the front slide of that, or the top slide of that. Do you know when test scores will be put in? I realize, yes, test scores will be in tonight. I apologize for that. And I will send an announcement out whenever they are up. I apologize. There's And I think that I apologized on Thursday for that. And so then I should have fixed it, but I did not. I apologize. Okay, so here we are talking about sugars. Now, when we talk about sugars, we've got two major classes. We've already gone through these a little bit, but reducing and non-reducing sugars. Now, if you look at these, we've got a free anomeric carbon on ribose, making it a reducing sugar. This right here would be bound anomeric carbon. This would be a free anomeric carbon. Remember, when you're looking for an anomeric carbon, you're looking at and translating that linear structure to the cyclic structure. And your anomeric carbon is the new chiral center. So this right here, we've got lactose, which is a dimer of galactose right here, and glucose right here. Now, sucrose, we've got two anomeric carbons that are both involved in a glycosidic linkage. So that's the first thing that I wanted to talk about with our um, cyclic sugars. Now, with that in mind, one of the things that I wanted to bring to your attention, though, was I wanted to point out the anomeric carbons and one of the important points about them. So, the important point is, and I'm going to draw with a white background, I'm going to draw glucose. Glucose looks like this. We've got our there's our six carbon CH2OH. Okay. Now, glucose, if you remember on carbon 2, OH is down, 3, OH is up, 4, OH is down. That makes glucose. Now, if you look, however, at your anomeric carbon, you might see glucose drawn like this, or you might see that OH group there instead pointed downward. Okay. This is glucose just as much as this is glucose. Now, what makes these two different sugars different from one another? Since they are both glucose, and typically when we talk about a sugar and OH group positioning, that's going to designate one sugar versus a different sugar. Well, when it comes to anomeric carbons and when it comes to cyclization of sugars, if you see an OH group pointed downward, that is known as the alpha configuration of this sugar. So this right here is alpha D glucose. Okay. Now, if that OH group is pointing upward, so I'm going to draw an arrow pointing upward, then it is beta D glucose. Now, the important thing about that is, and the reason for that and how this plays into an anomeric carbon is sugars can interconvert between the alpha and the beta, or the beta and the alpha configurations. And that interconversion is with or observed in a process known as rotation. Now, it's always helpful for me to think about reducing versus non-reducing. And whenever I think about reducing and non-reducing, I always tend to gravitate towards thinking about, well, reduction and oxidation, basically thinking about, oh, well, electrons are moving around. Now, This mutarotation, the interconversion from reducing sugar to, or sorry, a mutarotation is our interconversion from the alpha and beta configuration. One thing that I want you to know is that this OH group, it's not like it's on a pinwheel where this just swings up. Instead, in order for that, for a sugar to go from the alpha configuration to beta configuration, the ring opens up, opens and rearranges. Okay. So that's one of the things that I wanted to talk about with respect to alpha and beta configuration. The slide deck, somebody asked me, what slide deck are these from? These are from week 9, day 1. Week 9, day 1. Let me go ahead and then I got a private message. Can you explain what free means again? Yes, I can. Let me first get the link for our slide deck, and I think that I can pin that. Um, so, so I'll add the link to the chat. Copy link address. Everyone, done. And then, can I pin that? No, I'll just leave that there. Okay. Then free. What does it mean to be free? So let me go ahead and give myself a blank slide so I can work on that. There we are. New slide. There we go. Okay, so the question was posed to me of what do you mean whenever you say free carbon? Okay, so let me go ahead and show you. And I'm just going to draw a ink color black. There we go. Okay, so um, okay, OH, 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 OH. So this right here is alpha glucose. I'm pointing an arrow to my anomeric carbon. Now, whenever I say free, what I mean is that that anomeric carbon is bound to an OH group. That's kind of the biggest indicator that you have a reducing sugar. Now, I can make this a non-reducing sugar like this. OCH, 3, OH, 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 then CH2OH. So OCH3. Okay, so whenever I'm thinking this is a NR, whenever I'm looking at a sugar and I'm trying to determine if it's a reducing or a non-reducing sugar, I'm going to look for an oxygen within my ring. I'm going to find my anomeric carbon, and then if I have an OH group bound to that, then that's my indication that I have a reducing sugar. Now, if I have an oxygen within my ring, anomeric carbon, and then there's my oxygen that I'm singly bound to, but that is then singly bound to an R group, then I'm not dealing with a reducing sugar. Now, as an example of that, or as a kind of further example of that, we have this right here. CH2OH, done, 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 OH, 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 and then O, and then down, up, down, CH2OH, and then OH, OH, and OH. Okay, now what I want to do is draw your attention to this end right here. What I want you to focus in on is I want you to see that, okay, here's my oxygen within my ring. Here's my anomeric carbon. And then here is that oxygen that's bound to my anomeric carbon that is outside of my ring. Well, that oxygen is singly bound then to another sugar at position C4, as it would be. Okay. Now, what that means is that this end right here is not open. What that means is that whenever we have an anomeric carbon and we have a reducing sugar, what can happen is that these electrons right here will swing back to that, which will lead to these electrons going right here, so that we reopen the ring and we temporarily have C double bond O. For the person who asked that question, did that help at all? So to be considered open, it has to be reducing. Yes. Did that help? Okay. Now, let's look at this entire sugar down here. Let me ask you, how many anomeric carbons does this have? Add it to the chat. Add the total number of anomeric carbons that molecule has, the one that has the big black umbrella over it. Two. Perfect. Here's one. Here's the other. Okay. Now, just 1 second. Okay. All right. So I agree that there are two anomeric carbons there. Now, would you classify this sugar? And let me go through definitions. If you have um, one open end, let me erase that, then you do have a reducing sugar. So is this a reducing or a non-reducing sugar? Hit an R into the chat for reducing or an NR for non-reducing. Perfect. This is a reducing sugar because this end right here can open up. All right. Now, with that in mind, let's go ahead and go look at the rest of what we were going to tackle today. So I want you to be able to identify some select disaccharides. So a disaccharide is multiple, or it's two sugars put together. The disaccharides that I want you to be familiar with are sucrose, lactose, and maltose. This is sucrose. On the left-hand side, we've got glucose. On the right-hand side, we have fructose. Okay. Now, my question for you guys, when you look at this, or what's a valuable skill for you to be able to do, is to answer a couple of questions. One, is this a reducing sugar? And in order to answer that question, you've got to think about, okay, well, glucose, that's an aldose. Could you give me a Y or an N in the chat if you remember that? Okay. Fructose. Is that a ketose or an aldose? Add a K for ketose and A for aldose. Spot on. It is a ketose. Okay, so if you think about a ketose, you've got a structure that looks like this. There's carbon 1, carbon 2, carbon 3. Is everyone good with that? Give me a Y or an N in the chat if you remember that. Okay, so when you cyclize a ketose, chances are carbon number 2 is going to be your anomeric carbon. Carbon number 2 is going to have that carbonyl oxygen. And so what that means is C1 for your aldose is your anomeric carbon. That's C1. C2 is going to be your anomeric carbon for a ketose. So these two carbons are our anomeric carbons. Could everyone give me a Y or an N in the chat if yes, you see that, or an N if no, I didn't, I missed it on one of them. Okay. So we've got anomeric carbon to anomeric carbon. So would you classify this as a reducing R or an NR non-reducing sugar? And we know that, okay, from the vantage point of glucose, there's our oxygen within our ring, anomeric carbon going to, well, O rather than OH. So this end, I'm going to say it's closed. Now I'm going to go, Melanie, spot on. You're absolutely right. Because this is, actually, let me take it back a step. Now I'm going to circle it in red for the vantage point of glucose. Now I'm going to take a look at it from the perspective of my ketose. Here's the oxygen within my ring. Here's my anomeric carbon. And now I'm circling my oxygen that if I had a reducing sugar, that would be an OH. But from the standpoint of fructose, well, this is also closed. So this is a non-reducing sugar. Can everyone give me a 7 if you followed all of that? Okay. So this is our disaccharide of sucrose. It's glucose and fructose. We've got a non-reducing sugar here. The next thing, if you were to look at this, how would we classify this? Would we classify it as a beta or alpha sugar? And to answer that, what you want to do is you want to just look at that anomeric carbon right here and say, okay, which direction is my OH group, or would my OH group be going? If it is going down, then we have an alpha sugar. If it's going up, then we have a beta sugar. This is going down, so this is an alpha sugar. And I know that I kind of replaced, just to jump back a second, I replaced that OH right here with O and then another sugar altogether. Could everyone give me a 9 if they follow that, or a Z if, wait, what? Okay, so whenever we look at a sugar, what we want to do is just kind of what I always like to do. I'm going to erase some of this stuff. Is I like to say, okay, well, is this alpha or beta form of glucose? Okay, so I know that it's glucose, and what I'm going to do is just kind of temporarily scribble this out. Scribble out the other sugar of my disaccharide. I'm going to put an OH next to that oxygen that is found within my glycosidic linkage. And I'm going to see, oh, okay, well, this OH is pointed downward. So I'm temporarily putting there as a placeholder. Okay, now I'm going to erase that. Okay, glucose, fructose. Carbons, 1, 2, 3, 4. Da, 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 da. Now, this is a alpha 1, but it's a beta configuration from the vantage point of fructose. And that's because think about our directionality for our OH group of glucose, and then think about what we're looking at with respect to fructose. And I think the easiest thing to do if you're looking at fructose and you're trying to identify is this an alpha or a beta configuration, compare what direction the oxygen on our anomeric carbon is going compared to carbon number 6. The reason that we picked carbon number 6 is not some arbitrary selection, but instead, well, these things are going in the same direction. The same thing applies when you're classifying the sugar or classifying glucose as an alpha or a beta. Carbon 6 compared to the OH on carbon 1. If they are going in opposite directions, for instance, carbon 6 is up, carbon 1 is down, that is an alpha sugar. Carbon 6 is going down, and carbon 1 is going down. That OH group, those are both going down, then this is a beta sugar. Okay, so what I'd like to clarify with respect to that, just to avoid any sort of confusion, is 99 times out of 100. When you look at a sugar, if you look at, well, what direction is the OH on my anomeric carbon going? If it's going down, then it's an alpha sugar. Now, the remaining 1% of the time, what you always want to do is you want to compare it to, well, where is carbon number 6? So if I were to draw this, or sorry, to give you a different perspective on it, here is CH2OH. This is glucose. I'm going to erase that. Okay, could everyone give me an A in the chat if they see that that is glucose? Okay. Now, I have not designated this as an alpha or a beta. What I need to do is I need to recognize that, hey, most of the time, whenever somebody draws glucose, they've got carbon number 6 popping out up above the ring. Since this is inside the ring, that's going to be a good indication that, hey, I need to critically think about, well, where's my hydroxide group on my anomeric carbon? Okay, in this case, my hydroxide is pointing downward. But more importantly, it is pointing in the same direction as carbon 6. So C6 and OH on C1, same direction. Okay. So this right here takes precedence over the rule of, oh, if it's pointing down, it's alpha. So as, what on earth just happened? So as a result, how would we classify this right here? Is this an alpha or a beta sugar? Right, A for alpha, B for beta. Beta, perfect, great job. So when it comes to sugars, I would say more times than not, look at it and say, okay, the OH is down, it's probably alpha. But then kind of anticipate, okay, well, is the carbon 6, is that drawn inside the ring? Or where is it relative to my OH on my anomeric carbon? Okay, so my next sugar is sucrose. Sucrose is a disaccharide of glucose and fructose. Up at the top, we've got our glucose molecule. This is glucose in the alpha configuration, and then we've got fructose in the beta configuration. Again, looking at carbon number 6 and the OH on my anomeric carbon. Those are both going in the same direction, so that gives it a beta configuration. Carbon number 6 and glucose and the OH, or what would be the OH, those are pointed in the opposite directions. Therefore, that is how this is an alpha form of this sugar. Could everyone give me a 9 in the chat if they follow that, or a R in the chat if they are mixed up on that? Okay. Next up is lactose, which is a disaccharide made up of glucose and galactose. I'm going to say G and L. I'm going to circle a sugar. Tell me, is this an alpha or beta? And is it glucose or galactose? First, tell me if it's an alpha or beta, the sugar that I've circled there. Okay. Okay. Now tell me, is it G glucose or is it L galactose? Wonderful. Wonderful. Great job. This is galactose. So we've got galactose, we've got glucose. We have a beta 1,4 glycosidic linkage. Now, before I get into that, could you tell me, is this a reducing R or an NR non-reducing sugar? And think about our criteria for if it's a reducing sugar, we've got at least one open anomeric carbon. So when we look at this sugar right here, we've got a beta 1,4 glycosidic linkage. This glycosidic linkage, this is a closed end. So that is not a reducing end. Then if we look at glucose, the oxygen, the anomeric carbon, and then an OH. That's an open end. So this end right here is open. Therefore, this is a reducing sugar. This disaccharide is a reducing sugar. Now, yeah, okay. Next up is maltose. This is a disaccharide of glucose and glucose. Tell me, is this a reducing R or a non-reducing sugar? Yep, it is a reducing sugar because we have this open end over here. This has a alpha 1, carbon number 1, to 4, carbon number 4, glycosidic linkage. All right. So what I want you to be able to do whenever it comes to, I'm still not understanding reducing and non-reducing. Could you explain it one more time? Absolutely. Okay. So I'm going to erase all of this. And for anyone who is willing to answer, feel free. Okay. So when you think of reducing, think of reactive. Non-reducing, non-reactive. Okay. Now, in order for this group to be reactive, what we need is we need basically some electrons that, if we look at our oxygen that has the two single bonds, it's got two lone pairs of electrons. Then we have two lone pairs of electrons here. These electrons right here can jump over to this that hydrogen, or what we could do is we could have these electrons jump over here, and then this proton is lost. Okay. Now, we have basically the capacity, the ability for this whole thing right here, from this oxygen to that anomeric carbon to this oxygen, to open up. Now, these electrons being able to jump around makes this a reducing end. If a sugar has a reducing end or a reactive end, then it is considered or classified as a reducing sugar. Now, I'm going to draw a disaccharide of two glucose molecules where it is a non-reducing sugar. Okay, so first I've got to draw my first glucose molecule. And I'm just going to draw the six-membered ring. Got an OH, or yeah, an OH right here. Therefore, if that OH is pointing down, we are talking about the alpha form. And I'm really just bringing that up to kind of serve as a little bit of a reminder. Okay, now I'm erasing that hydrogen, and instead now I'm going to grab this yellow pen so I can kind of highlight the atoms that I care about. I care about that oxygen, that carbon, and this oxygen. So think about it almost like OCO. If that second oxygen, this one right here, has an OH, then I have a reducing end. But in this case, what I'm going to do is I'm going to draw a molecule that looks like this. It didn't turn out how I wanted it to. It's difficult to see that. So you could kind of treat this as answering the question, what am I looking for? I'm looking for OCOH. Does that, for the person who asked me to describe this again, Does that make sense? Looking for OCOH. Okay. When you look for that, your first O, so OCOH, your first O is the oxygen within your ring. Okay, so I'm going to look here. Check. Then I'm going to go to my anomeric carbon right here. Check. Last but not least, I'm looking for OH, a hydroxyl group. That would be right there. I don't have that. So I don't have a reducing sugar. Now, applying it to, so this is this sugar right here that I've just drawn. That's a non-reducing disaccharide. Because if I then kind of flip this and say, okay, well, I've got two anomeric carbons, I'm going to look at both of them. O, check. C, check. Now I'm looking again for OH. Do I see that OH? I don't. Does that make sense? Okay. Now I'm going to go back to my disaccharide of maltose, where I'm not connecting the two sugars through a glycosidic linkage. I'm going to say O, check. C, check. OH, no dice. Going back again to my other atoms. O, check. C, check. OH, done. Got it. This is a reducing end. Therefore, this is a reducing sugar. Does that help at all? Is the anomeric carbon always on C1 in the cyclic form? That's a fantastic question. And I'm going to go back just a tad to show you one example where no, it's not. When we have fructose, we've got fructose looks like this, CH2OH. I'm going to draw it separately. H, H, CH2OH, OH. So this is the cyclic form of our ketose, fructose. Is this a reducing sugar? Again, looking for the OCOH. O, check. C, OH, OH. So if we're looking to see if there is an OH at the end to determine if it's reducing sugar, yeah, basically that's all there is to it. Now, I want you to recognize, however, this kind of sequence, COH, is important because if you look at this oxygen, this carbon, but there's an OH there. Is that the same? Give me a Y or an N, anyone and everyone for that. No, it's not. Because this right here arguably is OCCOH. Does that difference make sense? Okay, great. Now, with what I was saying just a moment ago, your anomeric carbon, if you take a ketose, for instance, chances are your anomeric carbon is going to be carbon number 2. An aldose, it's going to be carbon number 1. You got to remember, where was the carbonyl oxygen? That's at the heart of this. Okay. Could you quickly go over how the directions of C1, C6 is different from identifying whether OH is just up or down, what it tells us? Okay, so, I hope that I answered this question. I'm just going to use this slide because I've got the space. So if we have CH2OH, I'm going to draw glucose. The way that I'm going to draw glucose more times than not is with carbon number 6 pointing out of the ring. Now, a fair question there is, well, why? Are you just doing that for the sake of the demonstration? No, I'm doing it because I think that it's a good representation of what that molecule looks like. Because can you imagine putting this group inside a ring? Well, if you did, you'd probably run into a bunch of steric hindrance there. So this is a good representation of what this molecule, or a better representation of what this molecule looks like. Now, this carbon 6 is pointed upward. Now, carbon number one, my anomeric carbon right here. At this point in time, I don't have a designation as to if this is an alpha or beta sugar. If I point my OH up, that's one piece of evidence that yes, it's a beta sugar. But more than anything else, this OH group and this CH2OH are both pointed, and just so you can kind of go into the same headspace as me, I'm looking at this ring as basically a flat plane. And so these two groups are pointed above my ring. Does that make sense? Give me a Y or an N in the chat. Okay. So since these are both above the table or above the plane, that makes this a beta sugar. Now, if I were to flip this, point it downward. Okay, again, that's a piece of evidence that, oh, well, they're going in opposite directions. More times than not, when you see the OH on carbon number one, my anomeric carbon pointing downward, well, that's an indication that I've got an alpha carbon. But you could, it might be easier for you to think about alpha is designed or is coming from C6 and anomer opposite. Beta is C6 and anomer same. Does that help? Does that make sense? So is it as simple as up, up is beta and down, up is alpha? Done. Yeah. Yeah, totally. Okay. Um, so as we proceed from here, now we're talking about polysaccharides. Give me a seven in the chat if you've ever heard of cellulose. Glycogen. Raise your hand if you like glycogen. Yep. Don't worry, you don't have to raise your hand in the chat. I can see you all doing it at home. I can't see you. Um, Glycogen, cellulose, and starch. These are all common polysaccharides. Cellulose is a structural polysaccharide. It's a structural polymer for plants. Now, one thing that I want you to notice, I'm going to scribble this one out, and I'm going to ask you this. I'm going to say this is carbon 2. Therefore, this is carbon 3. Therefore, this is carbon 4. Um, this is one of the things that you'll see from time to time in different models, in different pictures, that is kind of frustrating. Um, this bond right here, or this whole apparatus right there, is probably one that you're looking at and you're thinking, I've never seen a bond that looks like that. I've never seen a bond go and take like a sharp 45-degree angle and go straight up. I mean, if I think back to organic chemistry and I think about a point, what does that point usually indicate? A point usually indicates a carbon. So this depiction is one that you might see from time to time in different biochemistry textbooks. But this depiction is really just reserved for, hey, this is what this looks like. Or, hey, this is how we can kind of throw all this in there and have it be easily represented. Okay, so what I'm getting at the end of the day is kind of ignore this weird point there and instead think about this as an OH pointing straight down from carbon number 4. Now, with that said, if you have an OH at carbon 2 pointing down, an OH at carbon 3 pointing up, and an OH at carbon 4 pointing down, what sugar does that give you? 2 is down, 3 is up, 4 is down. Maybe down, up, down. Glucose. You're absolutely right. Okay. Now, I'm going to erase that and go back to this model. This right here is just a single bond. A single bond going to an oxygen and then connecting to carbon number 1. You might also see this depicted in this way. with like a curly line. Why is that? Well, it's because people are taking artistic liberties. Now, with that said, these two molecules, these are both glucose. Now, can you tell me, is this a beta or an alpha glucose? And it's tough to tell, but from carbon number one, we're going up, and then carbon six is up. So we've got a beta sugar there. We've got a beta form of glucose. So is everyone all right with the funkiness of this glycosidic linkage that connects my two sugars? Give me a Y or an N in the chat if you follow that, despite the weirdness. Okay, thank you very much. So cellulose is a bunch of glucoses. So if you go outside and get some bark off of a tree and start chewing on it, do you think any of the enzymes in glycolysis are going to be able to help you out and metabolize that and get all the glucose out of it and get all the ATP out of that? No. Why not? Well, because of this right here, the beta linkages. So cellulose is connected through beta 1,4 glycosidic linkages. Now, what's complicated about that is the enzymes in our bodies can't do anything with that. They're very stereoselective. So we can't break this down into individual glucose molecules. If we could, then boom, go eat some leaves and you're going to get some energy from it. Now, with that said, if we look at this molecule right here, or actually here, sorry, cellulose is connected by nothing but beta 1,4 glycosidic linkages. If we look at this molecule right here, glycogen, well, this is glucose, this is glucose. What's different about glycogen compared to cellulose? Cellulose is linear. It's just a single strand, a bunch of beta 1,4 glycosidic linkages. Glycogen, oh, well, it's got alpha 1,4 glycosidic linkages and alpha 1,6 glycosidic linkages, and that's it. Now, one of the things that's worth noting about glycogen is we've got one reducing end, and then these ends over here and here and here are known as non-reducing ends. So they're kind of the opposite of your reducing ends. So a single glycogen molecule is going to have one reducing end. Could everyone give me a Y in the chat if they follow that? That is a reducing end because I'm just going to draw in that OH right there. So that end is open. Can everyone see that? Specifically the person who asked. Okay, good. Now, a glycogen molecule is going to have one reducing end and then, well, multiple non-reducing ends. If we were to look at this and say, okay, can we deduce how many non-reducing ends? Here's one branch point, here's another branch point. And then here is the main chain. You could say for the non-reducing ends, you have one because there's going to be a main chain glucose plus the number of BP or branch points, BP. If you have two branch points, which is what we have here and here, 1, 2, each one of those has a non-reducing end. And then your main chain has a non-reducing end. Could you give me a Y or an N in the chat if that makes sense? Okay, great. So glycogen, what you need to know is alpha 1,4 and alpha 1,6 glycosidic linkages. One reducing end, multiple non-reducing ends, depending on how many branch points it has. This is your short-term energy storage. It's found in the liver and in muscles. This is the energy supply that people always like to point out to. Well, when you're running a marathon or some sort of endurance race, well, that's when you hit the wall is whenever your glycogen stores have been depleted. Now, With that, it's all made up of glucose, just as cellulose is all made up of glucoses. The difference is, are they alpha or beta linkages? Hmm. Okay, there we go. Now, last but not least of our polysaccharides is starch. This is a long-term energy storage molecule in plants. If you look at this, OH is down, OH is up, OH is down. Oh man, down, up, down. So this part of starch is amylose. This has a bunch of alpha 1,4 glycosidic linkages. The molecule shown here on the right is just kind of a continuation of that with the different representation. So amylose is a component of starch. So starch is multiple different sugars. One of those is a linear molecule known as amylose. Glucose, glucose, glucose. Amylopectin is your other component of starch. The difference between amylose and amylopectin is that, well, amylose is a straight chain, whereas amylopectin is a branched molecule. They're both made up of nothing but glucoses. But these two things kind of thread together to make starch. These are threaded together to make the more commonly known molecule known as starch. So these are, oh, great question. What about glycogen versus amylopectin? Well, one component of it is what this is showing is that your branch points between, so if you were to compare an amylopectin versus a glycogen molecule, what we have here is, let's just say that this is molecule number one. This is our non-reducing end. Or sorry, this is our reducing end. And then it has fewer branches, if I remember correctly. So these are every 30 to 50. So you'll see a branch once every 30 to 50 glucose subunits, whereas in glycogen, it's once every 7 to 11, uh, or 8 to 12, I guess it might be. So you're going to have a shorter gap between the branch points in glycogen than in amylopectin. Great question. Thank you for asking that. Okay. But here we go. Okay. Is everyone good on starch, glycogen, and cellulose? Does anyone have any questions about those three? Give me a Y or an N. Y if you're good, and N if you got questions. Wonderful. Okay. Now, if we proceed from there, there are other polysaccharides. Chitin, CHITIN, beta 1,4 glycosidic linkages, where at carbon number 2, you have an amine and an acetyl group. And these are found in exoskeletons of bugs. Think cellulose, but with N-acetylglucosamine, and it's beta 1,4 linkages. So N-acetylglucosamine instead of glucose. So if you were to ingest this, is your body really going to know what to do with it? It's not. Because of those beta linkages, it presents problems. In addition to that, the beta linkages for both cellulose and chitin allows for a more dense network of hydrogen bonding. Okay, so we've talked about alpha versus beta. We've talked about reducing and non-reducing ends. We've talked about disaccharides and polysaccharides and given some examples of all of these. Then chitin is one of our modified polysaccharides. Now, another thing that sugars are involved in is development. So, sometimes when people have, or when a person or an organism has a developmental disability or developmental abnormality, it's because of glycosylation. Now, anytime you see that ATION, I always kind of think about that as addition. Glycosyl or glycose, you're adding a sugar. So a protein can have a sugar added to it. Glycoproteins are proteins linked to oligosaccharides. Now, typically, an oligosaccharide is something that's a sugar that's about 5 to 7 sugars long. These sugars can be added, or they can be N-linked, which they are joined to asparagine in protein. This is done in the endoplasmic reticulum and the Golgi apparatus. They can be O-linked, joined to serine and threonine in protein, and this is done in the Golgi apparatus. Glycosaminoglycans are polyamino or polyanionic polymers of modified sugars, and peptidoglycans are peptides or proteins linked to polyanionic polymers of modified sugars. Okay, now what I want you to take away from this is I want you to think about a protein. Here's a protein, and that protein can be modified. Let's say there is a serine here. We know what a serine looks like. The serine R group looks like this. Okay. Now, that R group can be modified to place a sugar on there. Okay. Now, just for a moment, how would we classify this amino acid? How do we classify serine? That with the presence of that OH group, well, it can hydrogen bond, so we'd say it's polar. Okay. Now, with that OH group not really being an ionizable group, it's probably not going to have a negative charge. So would everyone agree this is a polar uncharged amino acid? Great. Now, with that single OH group, maybe that can jump around and do a couple of different hydrogen bonds. If, however, we added a sugar to that. So let's say that OH. I'm just going to draw a glucose molecule on it. Okay. Can everyone follow my actually we would have that gone. Everyone follow that there? Give me a Y or an N in the chat. Now, my follow-up question is, have I made, so this is like a serine with a glucose added to it. Has my serine with glucose added to it, is it more capable of hydrogen bonding or less? And look at what we got. There's an OH. There's an OH. There's another. There's another. We've gone from one OH group to now four. Does everyone follow that? So the abundance or number of hydrogen bonds that this molecule can take on, it's gone up. Give me an M if you follow that. Okay. So this is significant because now this amino acid and therefore this protein can interact with a wider range of things, a wider range of other proteins, a wider range of other organelles. Now, what that leads to is a kind of complicated network. So if you think that a protein structure is kind of bonkers, I drew it like this. Okay, there's our protein. And let's say that we have a serine on here. Serine just is a lowly OH group. Each one of these boxes represent sugars. If you think about the molecular weight of this, we can take a serine from a single OH group, which, yeah, it's going to mediate some hydrogen bonding, to 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17. 17 different sugars. Is that larger than this? Give me an L for larger and N for no. So I always kind of think about this, and I hope that this is helpful. Um, think about it like this protein right here wants to interact with another protein. And it is either going to go fishing with a night crawler, a little worm, or some sort of, or it's going to go fishing with a net. Which one do you think it's going to catch more fish with? The net or the worm? Probably the net. That's what this right here effectively is. It's going to be able to mediate more contacts with other proteins. Okay, so what I'm going to do now is we're going to switch gears because this next bit, this glycosaminoglycans, um, I'm not going to ask questions about that on your exam. This is, if you're curious about it, you're more than welcome to look into this. This is how another role that sugars can play in cellular processes as well as blood clotting and connective tissue. Um, but what we're going and in cell walls, we're going to instead change gears a little bit. And now we're going to talk about proteins and protein sequencing. So, does anyone have any questions about sugars or about like what do we need to know from what I just covered? Okay. So, changing gears a little bit to talk about proteins and analyzing proteins. We've talked about amino acid structures, we've talked about enzymes. Now we're talking about just sort of analyzing proteins as molecules and sequencing proteins. Has anyone ever heard of the Human Genome Project? This next slide deck is, thank you very much. Let me get this slide deck linked. Um, Copy link address. Is that right? Yeah, that should be right. Sequencing, copy link address. Okay, there we go. Yes, this is titled Week 10, Day 1. There is no Week 9, Day 2 slide deck because that was allocated for our exam. Okay, so when you talk about the Human Genome Project, that's invaluable because that gives you a human genome sequence, which enabled people to predict what a protein sequence was. And so for a time, it was almost like, well, do we really need to do we need to sequence proteins? How valuable is that? And the answer is it's still valuable. Because a sequenced protein gives you an idea of like, hey, this is the molecule that's present in a cell. This is what functions. This is how it functions. Um, it's very important to be able to analyze proteins. So, some pretty common questions that we're going to be able to answer about this. Some that you have some context for. We've used some of this terminology before. What is the sequence and composition? So, what are the amino acids that make up a protein and how are they organized? Which is the first one? Which is the last one? What is the 3D structure of a protein? How does a protein find its native fold? How does it achieve its biochemical role? How is its function regulated? How does it interact with other macromolecules? How is it related to other proteins localized within the cell? And what are some of the properties of it? Now, what we're going to get to today is we're going to start studying proteins and peptides. And in order to do that, you have to be able to purify it. Biochemistry is a very reductive field. You want to take a very complex mixture and distill it down to a small number of things. So polypeptides contain different amino acid sequences. Okay. The sequence and arrangement of amino acids gives the polypeptide a chemical character. Is that molecule charged, polar, hydrophobic, etc.? Some polypeptides bind specific targets, which can allow you to kind of fish those out and grab those out of there. So, whenever you talk about proteins, one of the important parts of protein analysis is purification. So, if you were to go to the grocery store, go to the butcher's section of the grocery store and get a steak. Um, would there be protein and proteins, different proteins in that steak? Yeah, absolutely. Um, are there sugars in that steak? Yeah. Is there like what is the diverse set of things that are in that steak? Is there fat in that steak? Yes, there is. You need to be able to take that complex mixture, that steak, and extract a protein or as much of a specific protein as possible. So, whenever you're doing a protein purification, you want to analyze your process and you want to analyze and make sure that what you're doing is effective. So, you want to look at what is your total protein concentration and compare that to your desired protein concentration. So, if anyone went trick-or-treating, chances are they got a bucket filled with lots of candy. And let's say that they only like green M&Ms. In their bucket of candy, they got a bag of Skittles. They've got M&Ms and then Reese's Pieces. And let's say just for the sake of the demonstration here, example, um, you got all of those candies and emptied them out. You got peanut M&Ms, regular M&Ms, Reese's Pieces, Reese's Pieces. You got your green M&Ms, the things that you want. And then you also got some Jawbreakers, some atomic fireballs, some warheads, some airheads, and then you got some blue M&Ms in there too. Okay, so let's say that you only wanted green M&Ms. This is kind of my example of protein purification and the process that it takes. Let me ask you, how many total proteins do you have? If each one of the candies represents an individual protein, how many total proteins do you have? Take a moment and count those up. I got 17. Yeah, Melanie, you and me both. 17 candies, 17 proteins. Okay, now how many of the candy that you want are in there? Three. So I'm going to say desired candy. Actually, I'm going to go ahead and say desired protein. Three. Okay. So you could look at this and say, all right, well, 3 out of 17. And when you're doing protein purification, you've got this same sort of calculation that's taking place. Um, and so you're going to go through a series of steps to try to enrich for or isolate just the green M&Ms. Now, let's say that we had, and some of these let's say some of them are peanut M&Ms. So the shape is going to be a little different. Let's say that what we did was here's one step after the purification. I'm going to go ahead and just kind of draw another set of candies. Did I make it? Okay, good. So I did a purification step that took me from this red bucket to the green bucket. Um, let's do an update. How many total proteins do I have? 6. How many of my desired proteins do I have? 2. So I've got 4 of the blue ones and then 2 of my desired proteins. Did I lose some of my desired protein? I did. I went from 3 proteins or 3 copies of it down to 2. Is that bad? Well, it's not great, but is it you know, was this step worthwhile is the question. And in order to answer that, What you really want to do is look at kind of the total protein compared to what you have. So when you started out, 3 out of 17 proteins. You could say that your protein in this mixture was, let's go ahead and say 3 divided by 17 times 100. My protein is about 17.6% pure. Does everyone see that? Now, after my first purification step, 2 divided by 6. Now my protein is 33% pure. Was this a, did this help me enrich for my protein? Give me a Y or an N to the chat. Yeah, I lost some of my protein, but I enriched for my protein. So anytime you do a purification step, that's the question that you want to ask. Did I enrich or purify for my protein? If the answer is yes, then fantastic. So if an enzyme, you want to be able to calculate that enzyme activity. Now, here's what that looks like. So let's go ahead and I'm going to erase all this. And here's some of the numbers that you might expect to see if you were doing a protein purification. I'm going to tell you about each one of these steps, and then we're going to extrapolate and expand upon these. So our very first step is a crude cellular extract. That would be basically like you take, let's take heart tissue, and you put it in a blender. You put it in a blender, and then after you've blended it in 1,400 milliliters or 1.4 liters, you quantify it, and you see that there is 10,000 milligrams of protein. It's a lot of protein. You are looking for an enzyme, and you see that you have 100,000 units of enzyme activity. Okay, so this is kind of like your starting point. This is effectively where you say, okay, well, there's 17 pieces of candy. I've got 3 of my desired candy. Okay, so this is where I start off. The next thing that you do is a purification step where you precipitate some things. Ammonium sulfate. Ammonium sulfate is a salt. NH4, 2, SO4. And what ammonium sulfate, the salt, does is it helps some proteins basically precipitate or get out of solution. Now, what we did, if you look at the volume, let's focus first on the volume. We went from 1.4 liters down to 280 milliliters. Okay, so we cut out, in terms of volume, we cut out about 80% of our volume. Our total protein, let's see, 10,000 milligrams down to 3,000 milligrams. We got rid of about 70% of our protein. Okay. And whenever I say our protein, I mean the total amount of protein. What about our enzyme? We went from 100,000 units of enzyme activity down to 96,000 units of enzyme activity. In terms of, answer this question, did we lose some of our enzyme, Y or N? We did. But if you wanted to quantify this, you could say that I got rid of 70% of the junk and I only lost 4% of my enzyme. Is that a reasonable trade-off? Probably. The next step that we did chromatography. Have you guys done chromatography in any capacity before? Okay. When did you do any sort of chromatography? Probably in a lab, maybe organic 1 or 2, where you separate based on, do you remember what you separate, what property you separated by or based on? Polarity. Great. Some molecules are polar, some molecules are nonpolar. Gives you a way to separate things. What do you think you would separate by if you're using ion exchange chromatography? If you were separating ions, maybe charge? One thing's positively charged, one thing's negatively charged. You could separate those two. Okay. Now, going from step 2 to step 3, we went from 280 milliliters down to 90 milliliters. So our volume got cut a whole lot. Then, in terms of our total protein, we went through from 3,000 milligrams down to 400 milligrams. Just a moment ago, I said that, well, to go from where we started, 10,000 milligrams, down to 400 milligrams of total protein, I think that would mean that we got rid of about, let's see, 400, but maybe about 99.6% of all of the protein in there. Does everyone follow me there? Give me a Y or an N if you follow that number. Okay, so we got rid of 99.6% of the protein. The question is, how much of our enzyme did we get rid of? If we got rid of 100% of our enzyme, that would have been a bad decision. But we got rid of, or we lost, you could say, about 20% of our enzyme. Does everyone follow that? And those numbers? So, arguably, that was kind of a good move. Our total protein number. What I want you to notice about our total protein is it's going to go down. Our activity, that's the indicator for our enzyme, that is going down as well. At the end of the day, what we are most interested in when it comes to enzymes is known as specific activity. If you look at your specific activity, it was 10 originally. By the time it's all done, we're at 15,000 units of enzyme per milligram of protein. What that means is that if you look at your total protein, 3 milligrams, we got rid of 99.97% of the protein. But what amount of our enzyme did we lose? We lost 55%. But what this means, what this high number right here means is we got rid of protein, lots of protein, but saved what mattered. So to use the candy analogy, this would be like saying your favorite M&Ms are the green M&Ms. Okay, I'm just going to color green M&Ms. Let's say that you have 1, 2, 3, 4, 5, 7 green M&Ms. All you want are green M&Ms. And somebody comes through and they say, well, I'm going to get rid of all the Skittles. You'd say, okay, cool, that's fine because I'm going to keep the green M&Ms in there because you can get rid of all the Skittles that you want. I don't like Skittles. I only want green M&Ms. So they then get rid of Skittles. And then the next thing that they're going to do is they're going to say, I'm going to get rid of all the Reese's Pieces. And you're going to say, that's fine with me because I don't like Reese's Pieces. And if you start out with 1 million pieces of candy and you get rid of these, maybe you're down to, let's say, 100,000 pieces of candy. At 100,000 pieces of candy, you've gotten rid of 90% of the candy. Great. After that, someone says, okay, well, after the Reese's Pieces and the Skittles are gone, now I'm going to get rid of all the, I've got a very good technique where I can get rid of blue M&Ms. Okay, cool. That's great. You're getting rid of these things in successive steps where you're just fishing things out, fishing things that you don't want in there to begin with. You're fishing those out. Does everyone follow that? Until at the very end, when you look at the jar and you say, oh, okay, well, yeah, I got rid of a lot of candy, but you know what? All that's left are 5 green M&Ms and 1 red Skittle that accidentally sifted through. And that's fine. Because what this means is most of what stayed is what I actually wanted. Okay, I'm going to go ahead and stop there. We're going to jump off with this stuff on Thursday. I've got office hours tomorrow. If anyone has any questions, I'm going to get your exam grades up tonight. I apologize for that. If anyone has any questions, you're more than welcome to hang around. Other than that, I hope you have a great Tuesday, great Wednesday, and I will see you guys on Thursday.